Eating something yummy like cake, chocolate, well, you already know that after your sugary treat is digested, insulin will be released by your body to balance out that blood sugar. But have you ever wondered what happens to insulin after this? I am here to discuss this with you. Insulin has a short half-life in the human body, approximately 4 to 6 minutes. The uptake, processing, and degradation of insulin by cells is a complex process with multiple intracellular pathways, but they are quite interesting, so don't worry. There are mainly three sites in your body that have the tools to break insulin down: your liver, kidney, and in case of pregnant women, the placenta. The liver is the key player here, contributing to 50% of this total metabolism. So let's zoom in and see what tools they have to catabolize the poor old insulin. There are two main enzyme systems at play here: the protease and glutathione insulin transhydrogenase, or simply known as insulinase. Insulinase is a bit unique than protease because besides being present in high concentration in liver and kidneys, it is also found in the skeletal muscles and placenta, aiding in the uptake of insulin by the cells and clearing it from the bloodstream. The question arises, how do these enzymes act? Well, this is the structure of insulin. Now, if you look here, insulinase enzyme act by breaking the sulfide bond between the A chain and the B chain. Reduced glutathione acts as a coenzyme for the transhydrogenase, donates the hydrogen atom for the reduction and itself thus converted to oxidized glutathione. This results in the splitting of insulin into two chains, which are then broken down further by proteolysis. The exact mechanism is still unknown. Let's go into the next section to learn how insulin receptors work and what role they have to play.